Hello all, good evening. Today I am going to talk about the biological methods which can be used to detect or study genes, or DNA or RNA. So there are three types of biological method. First is chromatography, second is electrophoresis, electrophoresis method, third is blotting technique. So I will discuss all these methods line by line. First I will discuss what is chromatography. So basically what happens in chromatography, we have a thymine, which is basically a radioactive element. So suppose if there is some DNA and I want to study that DNA, like what is the location of it. So this radioactive thymine will get attached to the part of that DNA and we can see what is the location of that part because thymine gets attached to that DNA and as thymine is radioactive, we can see from where the radioactive waves are coming. So that part basically radiates some radiation, so we can use it to see that GNA part, that DNA part. Second is electrophoresis. In electrophoresis, basically what happen, we will divide the uh, we will divide the DNA into several fragments, and because and its fragment will be divided into the several parts which will be having different mass. So we will give the charge to these fragments as we have given the charge to it. So we will put all the fragments on the gel, and because of this charge, this fragment will move because of the electromagnetic force. And because of the inertia, the fragment which is having less mass, it will move more. And as compared to the fragment which is having more mass. So we will get some type of pattern like this. So we can see that the fragment which is placed here, it has moved higher. If we compare to the fragment which has placed here, it will move less. So by this pattern, we can actually know like which DNA, like the position of the DNA, because different part will have different masses, and we can actually get an idea of what is the exact structure of the gene. Then third is blotting technique. In blotting technique, basically the process is like we make some DNA, which is the opposite DNA. If I explain, suppose there is DNA and I want to study this part. So there is complement DNA of it, which is opposite of it. So I will make that DNA. I will make probe with this DNA, and basically this part, this A, it will get attached here. It will get attached to this part only because this A is the complement of this. So we can track the location of this DNA. After the tracking, we can see where it is going, and we can actually get the location of our DNA which we are going to study, our subject DNA. We can basically get the location of it by tracking the location of our opposite DNA. So this is the last technique, blotting technique. It can be used for the identification of genetic mutation, like where the mutation has been happened. We can make the opposite DNA of it with a probe, and that DNA will go and attach to that part. Then we can use it for the personal identification or location of certain gene. So today we have studied about three methods. First, a chromatography, second, electrophoresis, and third, blotting technique. So that's all as of today. Thank you.